The basics of 3D bioprinting. 3D bioprinting is an additive manufacturing process where biomaterials, such as cells and growth factors, are combined to create tissue-like structures that mimic natural human or animal tissues. Here's how it works. 1. Pre-printing stage. Scientists begin by creating a digital model of the desired organ or tissue. They use images obtained from MRI or CT scans. Next, they feed this digital model into a specialized 3D printer. 2. Printing process. The 3D printer uses a material known as bioink to build the structure layer by layer. Bioink consists of cells, often in spheroid form, and other components necessary for tissue growth. Each layer is precisely deposited, and the printer gradually constructs the three-dimensional object. 3. Post-printing steps. After printing, the structure is incubated to allow cells to fuse and form a cohesive tissue. Researchers can then introduce additional factors, such as growth factors, to enhance tissue maturation. The promise and challenges. While 3D bioprinting holds immense promise, several challenges remain. 1. Complexity. Unlike traditional 3D printing, which operates in 2 dimensions, bioprinting involves the X, Y, and Z axes. Creating intricate vascular networks within organs remains a significant challenge. 2. Materials. Developing suitable bioinks that mimic natural tissues is crucial. Researchers experiment with various materials, including hydrogels and decellularized extracellular matrices. 3. Functionality. Ensuring that printed organs function like their natural counterparts is essential. For example, a printed heart must not only contract but also pump blood effectively. The road ahead. As technology advances, we can expect more breakthroughs in 3D bioprinting. Imagine a future where customized organs are printed on demand, revolutionizing transplantation medicine. While we're not there yet, the progress made so far is awe-inspiring. The numbers tell a story of innovation, determination, and hope. One day, we may print life-saving organs. 3D bioprinting, an advanced field within additive manufacturing, has made remarkable strides. Scientists are now exploring its potential to create living organs. Recently, Israeli researchers achieved a groundbreaking feat. They bioprinted a miniature human heart. The basics of 3D bioprinting. 3D bioprinting combines cells, growth factors, bioinks and biomaterials using techniques akin to 3D printing. The goal is to fabricate functional structures that were traditionally used for tissue engineering. However, interest has expanded to other applications, including biosensing and environmental remediation. The process. 1. Pre-bioprinting *. This initial step involves creating a model and selecting materials. Biopsy samples provide cells, which are multiplied. Tomographic reconstruction from CT or MRI scans generates 2D images. These images guide the printer to create a 3D structure. Cells are mixed with a special material that provides nutrients to keep them alive. No scaffold is needed for certain tissue fusion processes. 2. Bioprinting *. During bioprinting, living cells are integrated with biomaterials. Layer-by-layer deposition of cells bioinks results in complex, multifaceted tissues. Innovations range from extracellular matrix bioprinting to mixing cells with hydrogels. Scaffolds are also printed for joint and ligament regeneration. 3. Post-bioprinting *. After printing, the structure is further processed, evaluated, and prepared for specific applications. Applications and challenges. Tissue engineering *. Bioprinting aids in creating tissue and organ models for drug research and potential treatments. Clinical translation *. Despite progress, translating bioprinted constructs into clinical use faces challenges due to complexity and cell requirements for functional organs. Environmental remediation *. Bioprinting functional biofilms with microorganisms can facilitate pollutant removal. 3D bioprinting, an advanced field within additive manufacturing, has made remarkable strides. Scientists are now exploring its potential to create living organs. Recently, Israeli researchers achieved a groundbreaking feat. They bioprinted a miniature human heart. The basics of 3D bioprinting. 3D bioprinting combines cells, growth factors, bioinks and biomaterials using techniques akin to 3D printing. The goal is to fabricate functional structures that were traditionally used for tissue engineering. However, interest has expanded to other applications, including biosensing and environmental remediation. The process. 1. Pre-bioprinting *. This initial step involves creating a model and selecting materials. Biopsy samples provide cells, which are multiplied. Tomographic reconstruction from CT or MRI scans generates 2D images. These images guide the printer to create a 3D structure. Cells are mixed with a special material that provides nutrients to keep them alive. No scaffold is needed for certain tissue fusion processes. 2. Bioprinting *. During bioprinting, living cells are integrated with biomaterials. Layer-by-layer deposition of cells bioinks results in complex, multifaceted tissues. Innovations range from extracellular matrix bioprinting to mixing cells with hydrogels. Scaffolds are also printed for joint and ligament regeneration. 3. Post-bioprinting *. After printing, the structure is further processed, evaluated, and prepared for specific applications. Applications and challenges. Tissue engineering *. Bioprinting aids in creating tissue and organ models for drug research and potential treatments. Clinical translation *. Despite progress, translating bioprinted constructs into clinical use faces challenges due to complexity and cell requirements for functional organs. Environmental remediation *. Bioprinting functional biofilms with microorganisms can facilitate pollutant removal. When will we be able to print new organs on a 3D printer? Photo: Alexander Ryuman, TASS. Photo: Alexander Ryuman, TASS. Millions of people in the world are waiting in line for an organ transplant. There are 1.5 million people on the waiting list in China alone, 113,000 in the United States, of which, on average, 20 people die a day without waiting for a donor. A new kidney, the most in-demand organ, has to wait from 3 to 5 years. This problem can be solved by printing the necessary organs on special 3D printers. However, not earlier than in the next 10 years. Bioprinting technology. How and why are organs printed today? Video. RBC. How does it work? The principle is about the same as in conventional 3D printing. We get a three-dimensional object on a special printer. The first stage is enterprise. First, a digital model of the future organ or tissue is created. MRI or CT scans are used for this purpose. Then they print, layer by layer. This technology is called additive. Only instead of a conventional 3D printer, there is a special bioprinter, and instead of ink, biomaterials. These can be human stem cells, which in the body perform the role of any cells, pig collagen protein or cellular material based on algae. If the cells are alive, they are taken by biopsy and prepared in a bioreactor, until they multiply by division to the desired amount. During printing, the bioprinter polymerizes the cellular structure, that is, binds it using ultraviolet light, heating or cooling. The cell layers are bound using a hydrogel, organic or artificial. Then the resulting structure is placed in a biological medium, where it matures before transplantation. This is the longest stage. It can last several weeks. During this time, the structure stabilizes, and the cells are ready to perform their functions. Then the organ is transplanted and monitored for how it takes root. Bioprinting. How to print living organs on a 3D printer, video, RBC. In addition to the usual additive ones, there are other bioprinters. Some of them print collagen directly onto an open wound. This way you can quickly grow new skin, even in the field. In this case, the maturation post-printing stage is skipped. There are also printers that print in outer space, in zero gravity. In the future, they can be used on the ISS. The history of 3D printing. Who produces bioprinters and how much do they cost? There are more than 100 companies in the world that produce bioprinters for 3D printing. 39% of them are in the USA, 35% in Europe, of which more than half are in France and Germany, 17%, in Asia, 5%, in Latin America. In Russia, bioprinters are produced by 3D Bioprinting Solutions, which is also engaged in research in the field of bioprinting. The cheapest and most compact bioprinter, Tissue Scribe by American 3D Cultures, costs from $1,500. In second place is the Australian Rastrum from Inventia for $5,000. Ether bioprinter from the USA can be purchased from $9,000. The middle segment, from $10,000 and more, is represented by BioX from CELLINK, Sweden, RegMath 3D from Spanish RX1 and Canadian Aspect Biosystems. From $100,000. The 3D bioplotter of the German Envision Tech is worth it. The Russian Fabian 3D Bioprinting Solutions is even more expensive. Finally, the most expensive bioprinters, more than $200,000, are NovoGen MMX from Organovo, USA, and NGBR from Poietis, France. In addition to the cost of the printer, the printing process itself is also a plus of 15–20% of the price of the entire project. Obtaining the necessary cellular material will be even more expensive. Photo. New story. The economics of innovation. What does the first 3D printed village look like? Why are printed organs still not being transplanted? So far, the most successful experience has been the transplantation of cartilage tissues, those very ears to Chinese children. Small bones made of artificial cells are printed on a printer, and then covered with a layer. They are planned to be transplanted instead of a broken or damaged area, after which they will completely regenerate in 3 months. In the future, they want to use the technology for spinal injuries. The most promising direction is 3D printing of leather. In 5 years, they promise that this can be done directly on a person, on top of or instead of the damaged area. Skin and other tissues are printed from cancer patients' cells to test different therapy options. More complex organs, such as kidneys or hearts, have so far been printed only as prototypes or transplanted into mice, but not humans. Photo. Zuma TAS. Industry 4.0. In pursuit of youth and health, 3D technologies for beauty. In order for the organs to take root and function well in the human body, the patient's cells are taken, and then they divide until they are enough to print. There are whole institutes that create cell lines for bioprinting. But the problem is that cells have a division limit, after which they are no longer usable. Therefore, it is possible to print a model of the heart, but not in full size, that is, it is not suitable for human transplantation. The second problem is that the printed organ must function in conjunction with the rest of the body, digest food, secrete hormones, deliver blood and oxygen. A complex system of cells, tissues, nerves and blood vessels is responsible for all this. It has not yet been possible to reproduce it exactly. The software for bioprinters is also under development. To bring it to perfection, you need to process a large amount of medical, clinical, and statistical data. Finally, bioprinting technologies are not yet regulated in any way. All research must go through all stages of testing, including on humans, and then, obtaining patents. So far, experts predict the introduction of technologies no earlier than in 10 to 15 years. By that time, bioprinters and cellular materials will be widely available, and even the most remote regions will be able to use bioprinting.